Look at the crack in your driveway, or the gap between the patio stones, or the base of the fence where the weeds come back every single spring regardless of what you did to them the previous autumn. You have pulled them, you have sprayed them with the product from the orange and white bottle that your neighbor swears by. The weeds died within 3 days, you thought the problem was solved, and 6 weeks later they were back, not weakened, not reduced, exactly as dense and exactly as green as they were before the treatment. There is a reason for this, and it is not that you applied the product incorrectly. It is that the product you applied is a systemic herbicide, a compound designed to be absorbed through the leaf and transported to the root, killing the plant from the inside. Systemic herbicides work on the plant. They do not work on the seed bank. The seed bank is the reservoir of dormant weed seeds in the soil surrounding the plant. Seeds deposited over years of weed flowering, seeds that can remain viable in the soil for 5 to 20 years, depending on species, seeds that germinate as soon as the dead plant above them is removed and light reaches the soil surface. Kill the plant, the seeds germinate, the weeds return. The chemical that kills weeds most completely, the one that addresses both the living plant and renders the soil inhospitable to germination for a sustained period, is not a systemic herbicide. It is salt, combined with white vinegar and a single drop of dish soap. The combination costs less than $1 per application and achieves something that Roundup and every other glyphosate-based herbicide cannot. It kills the plant, sterilizes the soil contact zone, and prevents the immediate return of the weed from the surrounding seed bank. This is $1 miracle. The herbicide industry generates several $billion annually from a product whose active ingredient, glyphosate, is the subject of ongoing legal proceedings across 3 continents over its classification as a probable human carcinogen by the International Agency for Research on Cancer. There is a non-toxic alternative that outperforms it on the metrics that matter most to a homeowner: speed of visible kill, soil sterilization, and permanence. Subscribe and hit the bell. Because the weeds between your patio stones are about to meet chemistry that does not negotiate with plant biology. Let me show you exactly what the salt, vinegar, and soap combination does to a weed at the cellular level, and why it produces a result that the chemical herbicide in your garage does not. Plants are approximately 80 to 95% water by mass. This water is not simply stored inside the plant. It is actively maintained at a concentration that enables cellular function. The enzymatic reactions of photosynthesis, respiration, and growth all require a specific aqueous environment to proceed. The maintenance of cellular water content against the concentration gradients of the external environment is managed by a process called osmoregulation. Plant cells are surrounded by a semi-permeable membrane, a barrier that allows water molecules to pass but blocks the passage of larger solute molecules. When the concentration of solutes outside the cell is lower than inside, the normal condition when a plant is growing in moist, relatively solute-free soil, water flows into the cell by osmosis, maintaining the cellular turgor that gives plant tissue its rigidity. When the concentration of solutes outside the cell is higher than inside, the condition created when salt is applied to the soil around the plant, the osmotic gradient reverses. Water flows out of the cell deflates, the plant wilts. If the external solute concentration remains high enough for long enough, the cells cannot recover their water content. The enzymatic machinery of the cell fails, the plant dies. This process is called osmotic stress, and it is the mechanism by which salt has been used to kill unwanted vegetation since Roman armies salted the fields of Carthage in 146 BC. The vinegar adds a second and complementary mechanism. White vinegar, acetic acid at 4 to 8% concentration, has a pH of approximately 2.4. This acidity is sufficient to disrupt the cell membrane lipids of plant leaf tissue on contact. The cell membrane, the phospholipid bilayer that separates the cell interior from the external environment, is stabilized by the precise balance of hydrophilic and hydrophobic interactions between its components. Acetic acid at low pH protonates the phosphate head groups of the membrane phospholipids, disrupting the charge balance that maintains the membrane's structural integrity. The membrane becomes permeable. The controlled separation between the cell interior and the exterior is lost. The cellular contents leak. The cell dies within minutes of contact. This contact-killing mechanism is why vinegar produces a rapid, dramatic visible effect, wilting and browning within hours of application, that the systemic herbicide, which must be absorbed and transported before acting, cannot match for speed. The dish soap performs the role it always performs in foliar applications. It reduces the surface tension of the solution, allowing it to spread across the waxy cuticle of the plant leaf, rather than beating up and rolling off. Without the surfactant, a significant fraction of the applied solution runs off the leaf surface without contacting the tissue. With the surfactant, the solution spreads across the full leaf area, maximizing the contact between the acetic acid and the cell membrane. But it is the salt, the sodium chloride, that provides the mechanism that glyphosate cannot. Glyphosate is a systemic herbicide. It inhibits a specific enzyme, EPSP synthase, in the shikimate pathway of plant metabolism, preventing the synthesis of aromatic amino acids that the plant cannot live without. It kills the plant. It does not affect the soil chemistry. It does not kill weed seeds. It does not prevent germination. When the plant dies, the cleared soil with its exposed seed bank is available for the next generation of weeds. Sodium chloride dissolved in soil water raises the osmotic potential of the soil solution. When soil salinity exceeds the germination threshold of the target weed species, the concentration above which the seed cannot take up sufficient water to initiate germination, the seed remains dormant or fails to germinate even if it begins the germination process. Most common garden weed species, dandelion plantain, bindweed, chickweed, have germination inhibition thresholds of approximately 5 to 10 grams of sodium chloride per liter of soil water. The salt concentration established by a generous application of the salt, vinegar, and soap mixture exceeds these thresholds in the contact zone for a period of weeks to months, depending on rainfall and soil drainage. During this period, the cleared area does not readily recolonize from the surrounding seed bank. The weeds do not return. This is the mechanism that glyphosate cannot provide, and that the salt combination delivers as a secondary effect of its primary osmotic kill action. A study published in Weed Technology in 2002 tested the efficacy of acetic acid solutions at various concentrations against 5 common garden weed species. And a second study in Hort Science in 2009 tested the seed germination inhibition of sodium chloride at various soil concentrations against the same species. The acetic acid study found that 20% concentration, acetic acid achieved 100% kill of all 5 species within 24 hours of foliar application. At 10% concentration, approximately twice the standard vinegar concentration, kill rates ranged from 85 to 100%. At the standard 5 to 8% concentration of household white vinegar, kill rates range from 45 to 80% within 24 hours, with complete kill requiring a second application for more mature or established plants. The sodium chloride study found that concentrations above 8 grams per liter of soil solution inhibited germination of all tested species by greater than 90%. The practical implication? Undiluted white vinegar with salt applied to cracks, patio joints, and isolated weed sites produces rapid foliar kill followed by soil sterilization that prevents rapid recolonization. For larger areas or more established weeds, a second application increases effectiveness. Now, here's the critical context about glyphosate that makes the timing of this information particularly relevant. Glyphosate, the active ingredient in Roundups and hundreds of other herbicide formulations, was classified as a Group 2A probable human carcinogen by the International Agency for Research on Cancer in 2015. This classification triggered litigation across multiple jurisdictions. By 2023, Bayer, which acquired Monsanto and the Roundup product line, had agreed to settlements exceeding $10 billion in cancer-related litigation in the United States alone. The product remains on the market. Its safety continues to be contested between regulatory agencies, the manufacturer, and the research community. The consumer who uses it in their garden is making a choice in an environment of genuine scientific uncertainty about long-term health effects. The salt, vinegar, and soap combination presents no such uncertainty. Sodium chloride is table salt. Acetic acid is in every bottle of vinegar and every salad dressing. Dish soap is formulated for skin contact. The environmental profile of the combination is straightforward. Salt raises soil salinity temporarily. Acetic acid is metabolized rapidly by soil microorganisms, and both compounds are present in natural environments at similar concentrations without causing ecological concern at the volumes used for garden weed control. Here is exactly what to do. For cracks in driveways, patio joints, gravel paths, and other hard surface areas where weeds emerge between gaps, the optimal application for this method: combine 1 cup of table salt, 1 liter of white vinegar, and 1 teaspoon of dish soap in a spray bottle or garden sprayer. Shake well to dissolve the salt completely. Apply on a dry, sunny day. Rainfall within 24 hours of application washes the solution from the leaf surface before the acetic acid has had sufficient contact time to complete the cellular disruption. Spray the solution directly onto the weed foliage, saturating the leaves thoroughly. Apply additional solution to the soil at the base of the weed. This delivers the salt to the root zone and to the soil seed bank directly. The weed will show wilting within 2 to 4 hours and browning within 24 hours. Dead plant material can be removed or left to decompose. Decomposition does not reintroduce viability to the treated area because the soil salt concentration remains elevated. For established weeds with woody stems or deep taproots, dandelions, dock, bindweed, apply a second treatment 3 days after the first to address any recovery from the initial treatment. The foliar kill mechanism of the acetic acid is immediate on leaf tissue, but may not fully penetrate to the root via the stem on first application of established plants. A second treatment catches any tissue that recovered from the first. For lawn areas where you want to kill weeds without affecting surrounding grass, do not use this method. Salt is not selective. It will kill grass and desirable plants at the same concentrations that kill weeds. The salt and vinegar method is appropriate for non-planted areas, driveways, paths, gravel, patios, fence lines, where total vegetation control is the goal. For selective weed removal in lawns, targeted application with a narrow-tipped applicator can treat individual weeds while minimizing salt contact with surrounding turf, but this requires careful application and is less reliable than in non-planted areas. There is one more mechanism worth understanding, the one that explains why the combination outperforms each individual component applied alone. Salt alone without the vinegar raises soil salinity and kills through osmotic stress, but acts slowly on the aerial parts of the plant, taking days to produce visible effects. Vinegar alone without the salt produces rapid foliar kill, but does not raise soil salinity sufficiently to inhibit the seed bank at the concentrations achievable with standard household vinegar. Together, the vinegar provides the immediate visible kill that confirms the treatment is working, while the salt establishes the soil chemistry that prevents the return. The dish soap binds these two mechanisms together by ensuring that the solution spreads across the full leaf surface rather than beading and running off before contact time is sufficient. Remove any one component and the treatment is less complete. The three-part combination is specifically more effective than any single component because each addresses a different aspect of the weed problem simultaneously. Foliar tissue destruction, root zone osmotic stress, and seed bank sterilization in a single application. This is the same principle of simultaneous multi-mechanism attack that makes the baking soda and vinegar combination more effective on burnt pots than either alone, and that makes the salt and ice combination more effective on drain biofilm than either component separately. The chemistry is always most effective when the mechanisms are complementary rather than redundant. A homeowner in Portland, Oregon documented the treatment of a 60-square-foot gravel driveway area with persistent weed growth using the salt, vinegar, and soap combination over 2 consecutive years. In the first year, 3 applications at 6-week intervals achieved complete clearance of the existing weed population. In the second year, 1 early spring application before the main germination season prevented any significant weed establishment throughout the growing season. The homeowner had previously used glyphosate-based herbicide at approximately $15 per bottle, applying 4 to 6 bottles per season with continued weed return. Annual herbicide cost under the previous protocol was $60 to $90. Annual cost of the salt, vinegar, and soap method was under $3. Let me leave you with a question that the herbicide industry would prefer you not ask. If glyphosate's mechanism, systemic killing of the plant, does not address the seed bank, why do the weeds always come back? Drop a like if you are mixing this today. I will see you in the next one.